Okay, so we have this absorption spectrum. So we call this the absorbance spectrum. For A. And it turns out the absorbance spectrum for B looks like this. And the chlorophyll does not absorb very much light in the green or yellow area. It reflects that light, that sunlight, and so that's why plants are green. Now it turns out that in the fall, especially on the East Coast and in the north, you'll see a changing of the leaves, right? The leaves change color. And the reason why they're changing color is that this chlorophyll is being degraded somehow. It's being degraded. And now what you see are other pigments. We call these accessory pigments. And those pigments have a different absorption spectrum than the chlorophyll A and chlorophyll B. They tend to absorb here in this maybe 650 to 700 wavelength of light. I'm sorry, they tend to reflect that, and they tend to absorb a little bit closer to this area right here. So those different colors that you see in the fall, it's not to make us go, ah, that's beautiful. I mean, biology has made that in order for them to collect sunlight that is seen at those times of the year. So that's why we see this changing of the colors in the fall. Any questions about this? It's fascinating, right? It's all down to these molecules and what they absorb and what they reflect. That's what we're seeing. Okay, so let's talk a little bit about how the absorption actually takes place. I'm going to have to go to your book. So we're talking about chlorophyll A and chlorophyll B. Those are the major acceptors of the photon energy, which is coming from the sun. And let's just go ahead and take a look at what they actually look like. So this is on page 678 of your book. And what you have here is this sort of phytol side chain, and you have this sort of large molecule here. It looks a little bit like a heme, except it has a magnesium here in the middle. And there's just a couple of different slight changes between chlorophyll A and chlorophyll B. Chlorophyll A has a methyl group here. Chlorophyll B has an aldehyde group right here. So this is the major acceptor of the photons that will eventually transfer electrons. Okay. I'm going to make this as simple as possible in the beginning, and then as I go through it again, we'll get into a little bit more detail. So we'll almost like an onion, right? So an onion, we're going to like peel the onion further and further until we get to the real kernels of knowledge at the molecular level. Okay. So I want to show you what these. Before I show you this, let's show you this diagram here. So here we have something called a light harvesting complex. Okay, and by the way, these are all found in the thylakoid membranes. So we're talking about the thylakoid membranes here. Here we've got these a photon, which is going to hit the antenna pigment molecules. That's another word for chlorophyll. Okay, so don't get so excited about antenna pigment molecules. So that's actually chlorophyll, and the photons actually are accepted, and then the energy is accepted, and then you get this sort of transfer of energy. But eventually, what's key here is that you finally get an electron transfer, And this electron transfer from chlorophyll is what's going to start the electron transfer in order to produce energy, and also in order to move protons across the membrane of the thylakoid. Let's just briefly talk about this figure on page 679 of your book. So you probably know that molecules can accept photon energies, and when the photon energy is sufficient, you get an electron that gets excited to a higher orbital, higher electron orbital, and then that electron can get transferred to another molecule. So here we have one molecule, here we have another molecule, and as I said before, it's this transfer of the electron which starts the electron transport chain. So you have a couple of views of what's going on here with these, with this chlorophyll. Now I'm going to talk about something that's pretty important, and that is something called the Z scheme. The Z scheme. So this is going to be on page 862 of your book. Let's just, first of all, why is it called the Z scheme? It's called the Z scheme because if you actually take your book and move it into this direction, you see the letter Z, okay? So that's why it's called the Z scheme, okay? And let's remember, we are talking about the thylakoid membrane here. So this is the thylakoid membrane. This is where the action takes place. And what you have here is kind of like a reaction coordinate diagram, where if you were to plot the x-axis, but the y-axis is also very interesting. It is the redox potential. And you guys probably remember redox potential because we had talked about standard redox potentials when we talked about mitochondria and how to calculate free energy change from standard reduction potentials, right? Or standard redox potential. Standard reduction potential, same thing, okay? So redox potential, standard reduction potential, you can see that the units here are volts, okay? So we talked a lot about that. And we said last time that the more positive the value, the better able the molecule is to be able to capture electrons. Whereas the more negative the value, the better able the molecule is to give off electrons. So we talked about that. And you probably remember those values pretty well. Oxygen is like positive 0.82 volts, remember that? And then I think NADH was negative 0.32 volts. I even talked about that in the lecture. So here we have this sort of scale here, this redox potential here on the left-hand side, just to sort of orient you. And there are actually two different photosystems in the thylakoid membrane. And I'm going to concentrate right now on photosystem number 1. Okay, so let's concentrate on photosystem number 1. Okay, so photosystem number 1, you have a reaction center called P700. Why is it called P700? It's a weird name. It's called P700 because the wavelength of light that can excite electrons in photosystem 1 can be 700 nanometers or less. 700 nanometers or less. Okay? And so the photon gets absorbed by the reaction center, and then you get this excitation of an electron, and that electron gets transferred to another chlorophyll molecule. So you got this chlorophyll molecules. And actually, let me see if I got this right. Yeah, A0 and A1 are chlorophyll molecules. Let me just check my notes to make sure that's That's correct. Yeah, chlorophyll. Yeah, chlorophyll. Okay, A0 is a chlorophyll. And I'm sorry, A1 is actually something called phyloquinone. So A1 is actually phyloquinone. And this phyloquinone looks very similar to coenzyme Q. Okay? Very similar to coenzyme Q. So you got chlorophyll here, you got this coenzyme Q looking material here. Okay? And then you've got this FX, okay? And FX is actually a iron sulfur protein right here. So you have all these electrons that get transferred to this electron transport chain. And eventually 4 electrons get transferred, and you get something called the reduction of NADPH to NADP plus to NADPH. So 2 molecules of NADP plus get reduced to 2 molecules of NADPH. So if I have 2 molecules of NADP plus, how many hydride atoms can 2 molecules of NADP plus take? How many hydride atoms? 1 each. Very good. Very good. 1 each. So a total of 2. And how many electrons are going to be in 2 hydride atoms? 4. 4, right? Each hydride atom has 2, right? So 2 times 2 is 4. So you see these 4 electrons here? These 4 electrons are coming in the form of 2 hydride atoms that will reduce the NAD plus to NADPH. Okay, so that's this arm of the Z scheme. Okay, so think about this. We've lost 4 electrons from this P700. Okay, and you guys know that you cannot. It's hard to stay like, in order to sustain this system, in order for this system to accept another photon, you got to replace those 4 electrons that have been lost here. Okay, so where do they come from? So now we turn our attention to photosystem 2. So we're going to turn our attention to photosystem 2, and this reaction center is called P680. So P680. And it's called P680 because the nanometer wavelength of light that P680 can accept is 680 nanometers or less. Okay? Why don't I ever say more? Why couldn't this accept a higher wavelength of light? But I always say less. Why do I say more? Why couldn't I have said it's going to accept 680 nanometers or higher?
So it's off the spectrum.
Excuse me?
It's off the spectrum.
It's off the spectrum. Okay. Somewhat. Think about it in terms of energy.
Or give off less energy.
Exactly. It's the energy aspect, right? So you guys know that higher wavelengths of light are associated with lower energy. So you've got to have a minimum of 680 nanometers of light, or it could be a shorter wavelength of light because that's higher energy in order for these 4 electrons to be excited here. So essentially 4 chlorophylls are going to be excited here, and they're going to transfer their electrons through a series of electron transporters, okay? And during this process, during this part of the Z scheme, we're going to get 4 protons from the stroma. Now let me ask you guys a question. The stroma, is that inside the thylakoids or outside the thylakoids? Outside. That's outside the thylakoids. Very good. Very good. Okay? So it's outside the thylakoids. Okay, so those protons then get transferred across the thylakoid membrane, and they get placed into the thylakoid lumen. Okay? And in that process, you're going to actually be able to replace the 4 electrons that were lost here in the P700. Well, you're thinking, okay, Dr. Momand, that's a big deal. You basically have moved the problem from photosystem 1 to photosystem 2, but we still have 4 electrons here that were lacking in this P680. We need to replace those in order for this system not to just be a one-trick pony. Okay, we got to replace the 4 electrons here. And as I said last time in the overview of photosynthesis, those 4 electrons ultimately come from water. Okay, so it's water that's going to supply those 4 electrons. And so we have a very, very interesting protein here. I should say protein complex because it has a lot of polypeptides. It's very complicated. There's some metal ions in here as well. This is called the oxygen evolving complex, OEC. Okay? OEC, oxygen evolving complex. It's also known by another name known as the water splitting complex, the WSC. Okay? So WSC, OEC, those are the same thing. And so what happens is that, and I've said this before in previous lectures, is that the electrons get removed from the water and transferred to, we have this thing called YZ. That Y actually stands for a particular amino acid that has an abbreviation of Y. That's tyrosine. Tyrosine, the single amino acid code is Y. So it's one of these, it is a particular tyrosine that does the transfer of the electrons within the OEC to replace the electrons that were lost in the P680 reaction center. Okay, how many electrons can we get from a water molecule? How many electrons can we get from a water molecule?
2.
Very good. Exactly right. 2. Okay, so in order for this scheme to make sense, let me just go ahead and write that out for you guys. Okay. So I guess this can be split into O2 plus 2 protons plus 2 electrons. Okay. So that is the reaction. Okay. And of course, we need more than 2 electrons. We need to have 4 electrons in order for the balancing to take place. So all we do is we just say 2 H2O molecules go into O2 plus 4 protons plus 4 electrons. Okay?
Time.
It is time. Okay. All right. We will stop right here.